Are we live? Wonderful. So my name is Alex Abizaid. I am from São Paulo, Brazil, and I'm going to be the host for tonight. This symposium is sponsored by Med Alliance. You're going to be able to discuss very interesting new technology on drug-eluting balloons. And for that, we have three outstanding speakers representing United States of America with Ron Waxman, South America, Ricardo Costa, and Europe with Florin, who is, of course, discuss the newest data on Cyrolimus eluting balloon. And I'm sure that you're going to be super excited because there are a lot of points for discussions, including the debate between Cyrolimus versus paclitaxel, the current indications for DCB, bifurcations, small vessels, in-stent stenosis. So there is a lot for us to learn with these experts. And to help me with the discussion, we're going to have Anico Damonte, who doesn't need any introduction, Gaston Dussailand from Chile, and Marco Weinstein from Brazil. So very eclectic representatives from multiple continents. It's my pleasure to invite my old friend and good friend, Ron Waxman, who's been a great supporter for South America, for SOLACI, for the Brazilian Society for so many years. And he runs a very successful course in Washington called CRT. And I was a witness from the very first CRT that he put together. And after more than 2 decades, you see a huge success. And Ron is really the point of connection between science and practice. So Ron, the word is yours. So you're going to present the technology. We are all very excited to hear. Welcome.
Thank you very much. Going to try to share my screen and get the slide presentation. It may take some reason I cannot share it, so I have to find why this is happening.
You don't see the green bottle share screen underneath?
I do see the share the screen, but for some reason,
maybe the technicians put you in a different mode.
Yeah, let me just try to see. This is... No,
yes.
But you see the whiteboard, I don't see my presentation. Yes. Which sometimes happens with Zoom.
Try again. If not, we can have Ricardo, and then you can come back.
Just 1 second. I think we're getting close.
Yeah. Yes.
Okay. Sorry for the opening glitch, but. My friends and colleagues, I was actually asked to discuss when and why to use drug-eluting balloon and also the importance of sustained limus release. I'll share with you important information, but first, this is my disclosures. I am a consultant also to Med Alliance, which is relevant to this talk. So maybe we should take things in perspective because we're performing intervention nearly 60 years. And before even PCI, there was bypass surgery. After that, Brunzi came, started with POBA, bare metal stent, drug-eluting stent, first generation, second generation, and then came a drug-coated balloon. And initially we had the first generation. And following that, there was the introduction of BVS. And what was common to both of those technologies were that they made the premise of leaving nothing behind. But as of today, we are discussing about a second generation of drug-coated balloons. We're moving again forward, and this is the Limus drug, which I'll try to convince you it's a more effective sustained Limus release, DES, like SLR. So there are a lot of applications for drug-coated balloon, and all of them are true also for the solution SLR. Peripheral SFA below the knee. In the coronary, the indication that are most popular, and this is how we started, was in-stent restenosis, small vessel, and then bifurcations, side branches, lesions. And then there are some indications for arteriovenous fistula with reduction of hemodialysis, shunt restenosis, which is very devastating for those patients, and keeping the dialysis access open longer. And finally, a very interesting application that has been tested or studied in the past with drug-eluting stent, but I think it's more appealing for drug-coated balloon, and this is erectile dysfunction. The over 300 million men worldwide having ED, and again, it's another interesting indication, not just to go with POBA, but with drug-coated balloon or drug-eluting balloon. So what is the rationale to use it in coronaries? I think we mentioned leave nothing behind concept. If we could, we would have left no metal in the arteries. And that would allow us to preserve physiological vasomotion, allow the vessel to grow, no caging of the vessel, things that we heard in the past with BBS. But I think drug-coated balloon or drug-eluting balloon offer the same thing, and it's overcome problems associated with the use of the permanent metallic stents, such as jailing of side branches, overhanging osteal lesions, inability to graft the stented segment. So many drawbacks of the stents can be achieved if we do drug-coated balloon and leave nothing behind. And then again, if you need to bypass that vessel, you don't go with a full metal jacket and allows grafting by having treatment just with drug-coated balloon. The absence of any residual foreign material and restoration of functional endothelial coverage can also reduce the risk of stent thrombosis. If we're not going to have a stent, we're not going to have stent thrombosis. And also, we're not going to have any stent allergy. And finally, there is no need for long-term dual antiplatelet therapy. So for all those arguments, we are actually enthused more and more about the potential of drug-eluting balloon or DCB. Today, I'm going to introduce to you a relatively latecomer in the whole drug-eluting balloon technology. It is the solution SLR, which stands for Sustained Limus Release, which is manufactured by Med-Alliance. So first of all, the first question is why Cyrolimus? We've been using paclitaxel until now, and results were, I would say, satisfactory and acceptable. But if you look overall and you see on the cell cycle, where is paclitaxel? Paclitaxel is in the M phase, and it does give you a relatively short window of therapy. Cyrolimus, however, is cytostatic, and it gives you also a immunosuppressive impact and anti-migratory agent that stops the activated smooth muscle cells to create a restenosis. And it does maintain the cells in the G0 resting phase and limits apoptosis. So overall, it's effective drug, which have been shown immensely on the drug-eluting. And not only that, it is a safe drug. It does give us a very wide range of safety in respective to the dose. So we are going to have this discussion of Cyrolimus versus paclitaxel. And you can see with respect to toxic effects, they are minimal with Cyrolimus compared to paclitaxel. And the therapeutic range is also different when you compare between the two, but I pay attention more to the window of therapy and the toxic effects, which are really different between the two. One thing that we have seen is solid clinical evidence based about Cyrolimus and anti-restenotic, the drug of choice. And when you compare studies, and you have a list of studies here, in each of one of them, when the Cyrolimus was compared to paclitaxel, within the drug-eluting stent, lead loss was lower, the TLR was lower, it was just simply more effective drug to put on the stent. If that's the case, it probably should be also true on the balloon. And indeed, the Cyrolimus has proven superiority over paclitaxel in also large randomized DES trials. And as a result of that, we are not having any more paclitaxel-eluting stents. We just have drug-eluting stent. But if you compare and contrast between the two, again, the mode of action is different. Sirolimus is cytostatic. Paclitaxel is cytotoxic. The margin of safety is 10,000-fold versus 100-fold. There is a wide therapeutic range versus narrow therapeutic range. It's both work for restenosis. Sirolimus also has some anti-inflammatory features, and it was accepted completely by the market. It does have a slow tissue absorption compared to fast tissue absorption with paclitaxel, and short retention compared to long retention with paclitaxel. So if you look at the last two features, there seems to be the drawback of sirolimus. And that's maybe the reason why we have not seen until now the technology converting like they did very fast in the stent era from paclitaxel to sirolimus. Because in order to turn those two red in the sirolimus to green, you have to have a unique technology. Again, paclitaxel is absorbed quickly and tends to localize in the subintimal space and partition significantly in the adventitia. Sirolimus, however, absorbs slowly and spread throughout the entire artery where it dilutes down to subtherapeutic level. So again, looking on both of them, there are definitely challenges with sirolimus. It is a cytostatic drug. It is safer. It offers unique anti-inflammatory, but there are the challenges. And what are the challenges? The challenges that overcame by or need to be overcome by sirolimus is to enhance the tissue absorption, to facilitate sirolimus enter into the arterial wall, extend the sirolimus tissue retention, which is less lipophilic compared to paclitaxel. So you'll cover the entire restenosis cascade, and also develop a new, more durable and washout resistant coating. So these are the main features that needs to be overcome if one wants to use a sirolimus eluting balloon. So this is being resolved by the proprietary technology of the SELUTION SLR, which is a Med-Alliance product. And what this technology offers you? It offers you a proprietary micro-reservoir technology, which creating micro-reservoirs that combining sirolimus and biodegradable polymer. Then you have the miniature drug delivery system, which is optimal size micro-reservoirs to achieve pharmacokinetic release profile, comparable to the best in-class DES, and consistent and predictable drug release that sustained therapeutic effect up to 90 days, and that should be sufficient to abolish restenosis. And the third component, which is very unique, is what is called CAT, Cell Adherent Technology, which is a proprietary amphipathic lipid technology which binds the microreservoirs to the balloon surface. And this contains and protects the microreservoirs during insertion and inflation. It enhances drug retention and the bioavailability, allowing for a lower drug dose concentration of the balloon surface, and optimize the transfer of the microreservoirs to the tissue and maximizes the cellular uptake of sirolimus. So again, just maybe more in details, this is the microreservoirs. They are precision engineered drug delivery system made of biodegradable polymer intermixed with sirolimus drug. Then you have the mechanical, as the balloon expands in one way, the biological highly lipophilic CAT coating binds to the fatty cells in the vessel wall. And then you have the third component, which is the electrostatic. These are basically the three mechanical ways in which you overcome the drawback of sirolimus and make it effective. Mechanical gives you the balloon compliance to ensure optimal apposition of the CAT coating to the vessel wall. The electrostatic is leading to the CAT coating that is attracted to the vessel through the ionic interaction. And finally, the biological, which is a highly lipophilic CAT coating that binds to the fatty cells and optimizing the microreservoirs to transfer into the vessel wall. These are some information that was done by Renu Vermani that shows very nicely the pharmacokinetics and the drug dose per balloon size, and also the arterial tissue drug concentration when you're looking here on the Cyrolimus versus the competitors. So for example, if you look here on the orange, the Cyrolimus, you see here that in 1 hour, the tissue drug concentration in microgram would be 262, and this is much higher than what you would see compared to the paclitaxel by 2 other competitors. And this is true all the way up to 60 days. And this is the electron microscopy that shown very nicely how this is a interface within 24 hours into the adventitia and the vessel wall. These are showing us, again, the concentration in the arterial surface. And when you compare the solution here in the green to the Xience, which is a drug-eluting stent, which we all know, it's very similar. If you take and compare it to the Medic Charge, which is another drug-eluting balloon with Cyrolimus, you see the superiority of the solution SLR over the Magic Touch. So what you're seeing here, that overall, if we look at this, we see that the solution SLR tissue concentration is very similar to what we're seeing with drug-eluting stent. And here, again, the drug transfer, comparing the Med-Align solution to the paclitaxel and to the Bibard and Medtronic, the Lutonix and the Impact. And this is a very important information. As you know, when you deliver the balloon to the target, you have a loss of material during the procedure, during the transfer to the target area. You can see here, the transfer here in the red is the smollett with the solution compared to the paclitaxel. More retained on the balloon and more transferred to the vessel. So in essence, we can claim here the efficacy of the solution would be superior to the one in the PTX balloon, at least by the delivery system. We have all this information. So the sustained release really does matter. We do need a longer elution than a short elution in order to overcome the entire cascade of the restenosis, because it takes time to complete the restenosis process. So the longer elution, the better. And there are some examples with what happens if you don't have it. And these are being taken from the drug-eluting stent era. So for example, Endeavor was inferior to Cypher and Xience because, again, the elution profile was different. If you look at the periphery, there was a difference between slower elution and fast elution with Cyrolimus, and you saw that this was impacting on the late loss. When you have a slower elution, it's 0.39. When you have a fast elution, it's 0.72 late loss. So obviously you'd like to see more slower elution, and that's what the sustained release offered. So the higher percentage of drug crystals in coating is associated with the higher tissue concentration through 90 days in the porcine arterial model. Again, the work that was done in Renu Vermani lab. And in the most efficacious paclitaxel DCB for the SFA, there is long-term presence of drug in tissue through the 90 days. Another important aspect of drug-eluting balloon, the drug-coated balloon, is the integrity of the coating. We see flakes coming out from different balloons. You can see them very easily if you do those in vitro. This can happen even when you just inflate the balloon. But for the Lutonix, for the solution balloon, you don't see it. So you can see the coating here on company A on the top and company B, but with solution SLR, we don't see any flaking. And finally, one word about the microreservoir technology, which called also microspheres. They have been extensively used also in other pharma industries, so it's not something new. And it's solve manufacturing problems that create extended release profiles and reduce toxicities, otherwise rapidly absorbed drugs. It is a very accepted technology, and unlike nanoparticles, microspheres can be precisely manufactured to an exact and repeatable particle size and elution profile, which give you more uniformity and predictable outcome. And it is a very known and effective technology to deliver drug therapies. From here, I'd like just to mention that there is a very robust coronary trial program within the SELUTION. Started with the first in man, going to the ISR IDE for in-stent restenosis. This is going to be a pivotal study for the US to get approval with 418 patients. Study that already launched in Europe, and we're waiting in the US to start it as well. Then you have the SELUTION de novo, which is the mega study in our field, 3,326 patients. That's going to be in Asia and Europe. And then the SELUTION de novo IDE in the US, which is currently under review. So a very robust program to support the data for the use of this SELUTION product. Finally, I'd like to just go over the study of the de novo study design. This is, I think, a very attractive. I think beyond in-stent restenosis, we claim that every place that you put a stent, you potentially can treat that also with drug-eluting balloon. So you have to screen the patients, you see if the patients are candidate, and then the randomization would be all DEB strategy versus all DES strategy. On the DEB treatment, it would be according to the hospital practice, including adequate vessel preparation. And again, for the DES treatment, it's going to be also according to hospital practice. So this one-to-one randomization of 3,326 number of sites would be really the first and the large one in this field. The PI of the SELUTION de novo study are Simon Eckestel and Christian Spalding from the UK and France. The objective would be to demonstrate non-inferiority for TVF of a treatment strategy with the SELUTION SLR DEB plus provisional DS versus systematic treatment with DES for the treatment of the novel lesions. The design is prospective randomized clinical trial. It's open label comparing the SELUTION DEB to DES. I told you about the number of patients, number of sites. The primary endpoint is TVF, cardiac death, target vessel related MI at 1 year, and the patients will be followed all the way up to 5 years, and TVF at 5 years would be also a co-primary endpoint. And then you have all the traditional secondary endpoint at 2 years, 3 years, and 4 years, and follow-up goes all the way up to 5 years. So this is really a pivotal study that can change the field, because if we do find, and this is the study question, the hypothesis, that indeed the SELUTION would be non-inferior to stents, I think that can bring us back to the balloon and revolutionize the way that we perform intervention. So to summary and the take-home message, I think we do recognize now that DEB offers the leave nothing behind concept. Preservation of physiological vasomotion and overcome problems associated with the use of permanent metallic stents. Cyrolimus is the preferred drug for DEB technology. The main challenges are tissue absorbance and drug retention. But the SELUTION SLR offers sustained release via proprietary microreservoirs and cell adherent technology. And the success of this technology in the field and the confidence of the sponsor on the product led to the design of a mega pivotal trial for ISR and the lower lesions. And I'd like to thank you for the opportunity, and looking forward to see the results of those clinical trials in a few years. Thank you.
Thank you so much, Ron. This was outstanding. I think we all learned so much, and this promising technology can really expand the indication for DCB. So we'll save the discussion for later, and I would like to invite Aníbal to present the next speaker. I'm sure that Ricardo already there, ready for his 10-minute talk. Aníbal.
For sure. Thanks, Alex. Thanks, Alex, so much. And I will introduce my good friend and the president of the Brazilian Society of Interventional Cardiology, a very active member of SOLACI, a very active clinical investigator, Dr. Ricardo Costa, who will present us the SOLUTION Sustained Lesions Release Clinical Data Overview. Thank you, Ricardo, and we are looking forward to your presentation.
Thank you, Aníbal, for the kind words, and thank you, Alex. It's great to be here, and after this great talk from Ron, giving why we should consider this new technology as something really to improve the outcomes. I'm going to bring the results of the first-in-man trial, the Cyrolimus, or the SELUTION Cyrolimus drug-eluting balloon. So the objective of this study was to assess the safety and efficacy as a first-in-man study of this device, and it did include de novo and in-stem stenosis lesions in vessels 2.0 to 3.0 millimeters in diameter and lesion length less than or equal to 18 millimeters. This trial considered at least 50 patients in 6 clinical sites in Asia, and the primary endpoint was freedom from device and procedure-related mortality at 30 days, and then all of the common angiographic and clinical parameters at 6 and 12 months. In terms of the trial oversight, we have here Stefan Windecker was the chairman, along with Don Cutlip and Robert Byrne, all known in this field. And we had the core lab, graphic core lab at the CRC in São Paulo, Brazil. Overall, there were 56 patients, 1 patient and 1 lesion included. There was 1 case, there was a bailout stenting in a side branch that was pre-dilated, was stent implanted in the main vessel, ended up receiving a stent, so we did not include in the main results. Overall, there are 55 considered for angiographic analysis, and the 30-day clinical outcome was completed in all the cases for the primary endpoint and the 6-month follow-up in 44 cases. The enrollment was between 2018 and 2019. And here you can see a summary of the key clinical profile of these patients. 50% diabetics. This is like what we see in some countries in Asia. Smoking 27%, previous MI 14%, and previous PCI 29%. In terms of pre-procedure QCA, the mean lesion length was 11 millimeters, the reference vessel diameter 2.21, and the percent stenosis around 68%. At post-procedure, we can see an acute gain of 1.07 and a balloon artery ratio around 1.1, which we consider to be an optimal outcome for these balloon angioplasty cases. Out of hospital clinical events, we did not have any events up to 1 month. So the primary endpoint, which was freedom from device and procedure-related mortality through 30 days, we could consider 100%. And at 6 months, there was 1 target lesion revascularization that was, we're gonna also show this case. In terms of the technique, balloon predilatation as recommended by the protocol was performed 82%. The study drug-coated balloon was performed in all the cases. An additional balloon was used in 2 cases, 4%, and bailout stent implantation was done in 1 case. Here we can see the community distribution CFD curves for MLD, and in the far left side, pre-procedure 0.71, at post 1.79, at follow-up 1.53. Here the community distribution curve for percent diameter stenosis. We can see here the pre-procedure, and then at post and follow-up, we don't see a major difference between this important parameter for this type of device. And here the community distribution curve for late lumen loss. Most of them, you can see a more narrow distribution to the left side, and a few outliers, as you can see plotted in this graph. The mean values at 6-month QCA, the percent diameter stenosis was 30.5%, and the late lumen loss was 0.26%. Of course, this parameter was performed in those with pair angiographic analysis. I bring you a few examples. Perhaps this is the most considered criteria for DCB to date, which is in-stent restenosis. So we can see here in the prox LAD, a very severe subocclusive lesion at pre-procedure. Then after dilatation with 2 drug-coated balloons, solution drug-coated balloons, we can see the follow-up, just a steady, perfect result from post to 6 months. Another example, a very similar lesion, also subocclusive ISR in the proximal LAD, and at post-procedure and follow-up, a great, well-sustained result. Here we can see another well-represented type of lesion in this study, which was small vessels, and a more distal circumflex at pre-procedure, very severe stenosis, at post-procedure, and then at follow-up, just a great result as well appreciated in these illustrations. Another important subset that's being more and more considered for drug-coated balloons, a side branch, bifurcation side branch. You can actually simplify procedures with this type of device. And at pre-procedure, you see osteostenosis. At post-procedure, after having stent in the main vessel, and here at follow-up, a great, well-sustained results. And here I bring you the only major adverse clinical event reported. This was a subocclusive ISR to begin with. And importantly, this patient, which was not, procedure was not done as recommended by the protocol, he first had a paclitaxel DCB to predilate his lesion. Then following, he did have the study device, and this was a post-procedure result. And this patient came back with a very severe stenosis, and it was the highest late lumen loss, 2.14, and this patient underwent TLR with a regular metallic stent. So with that, I would like to conclude for this 10-minute presentation that the solution Cyrolimus eluting balloon with a sustained release demonstrated favorable clinical and geographic follow-up results, and these results have supported the CE-mark submission and approval of this device. Thank you for your attention.
Ricardo, listen, this was great. And again, it's good to see that this new technology is not only a theory, there is some clinical results, and we are very excited to see how it's going to evolve with larger trials. So our last speaker is Florin Cuculi. He's from Switzerland, and we have to really recognize his efforts. It's already tomorrow in Switzerland, so it's 1 a.m. in the morning. So thank you so much, Florin, for joining us and to contribute to SOLACI. The word is yours. I'm sure that you're going to put some pepper in this presentation in terms of how we can push this technology in clinical practice. Thank you so much.
Thanks a lot for your very kind introduction and the possibility to speak to you. I'm here with my son, who is 10 and didn't want to go to bed. We are actually on holiday, so that's why I could afford to stay up this long. So what I would like to speak, I would like to go more into technical details, because we have been using the solution balloon extensively in my lab. We have probably used more than 400 balloons so far, and most of them in native lesions. I would like to speak more how to prepare lesions, and I will show you some clinical examples, and I'm very excited to have then maybe some discussions. I mean, to start with, we all know the career of our patients. They start with stents, they go for bypass, and they come back for stents, and they receive balloons, and then they receive more stents, and then we put the coronary sinus reducer because we have nothing better to do with them. And we have, as Ron also showed, we have the problem of stent thrombosis. This is rare, but if it happens like this in this patient, then it's quite detrimental for the patient, because this patient is now in heart failure. And we have also this problem, that the vasoconstriction, which we see from time to time. This was a patient who was extensively stented in the past, and I was able to catch this severe vasoconstriction, as you can see very distally in the LAD, which could be corrected with repetitive doses of nitroglycerin. Now, if you look why stents fail, then we know we have some factors: diabetes, CTO, ISR, long lesions, and small diameter. And I think this is exactly the situation where we should be thinking about using drug-eluting balloons, because we know stents do perform badly in this kind of situations. And this is maybe, let's say, the ground for my talk, because I try to use drug-eluting balloons, especially SELUTION, exactly in this kind of situation. And I do not make exceptions in calcified lesions, because I think in calcified lesions we know even better than stents perform badly. So if you can replace stents with balloons in such situations, why not? Let's do it. And this is a case which was not treated with a balloon, but which made me discover a new way of preparing lesions. This was a calcified lesion, and I said, why don't we just cut and crack it? So why don't I use first a cutting balloon? I use usually Wolverine to cut the calcium, and then I used an OPNNC balloon to crack the calcium and create lumen. I used this many times for lesion preparation in calcified lesions, and then I discovered that actually this is a good way to even treat normal lesions if I want to treat the lesion with a drug-eluting balloon. So all the cases I will present are cut and crack and then SELUTION cases. Now, you will have the situation of a patient who is 38 years old with familiar dyslipidemia, and I think putting a stent in this patient is just the start of his end, because this is, we know they will look great in the beginning, and he will start to have late lumen loss, maybe because this is a high-risk patient. But in this case, it's actually a bifurcation. So I put this case on LinkedIn, and then most of my colleagues suggested to use 2 stents, in fact, in this kind of patient. So use 2 stents for a bifurcation in a 30-year-old man. I think it's wise, and this is what we have done so far. But since in the introduction of SELUTION and some really early encouraging results, I said, well, I'm going to try to avoid it. So in this case, I use a Wolverine balloon to cut that lesion, especially in the main branch. I use a smaller balloon for the side branch. Then I use a 3O balloon to create some lumen. OPNNC, it's a high pressure balloon, but I use it at lower pressure because I don't want to completely dissect the vessel, but really just expand the lesion and gain some lumen. Then I did some casing balloon inflation before I used the SELUTION balloon, and then I used the SELUTION balloon. And I will re-angio this patient in the future. But you can see this is what the result looked like. And I left it like this, and this is now about 4 months over, and the patient has not come back, and he hasn't had a stent thrombosis. Of course, angiographically it would look better if you do it with 2 stents in the initial phase, but the patient is symptom-free, and he's 38, and we still have enough time for this patient to put some stents there. Now, I also showed you that in CTO, stents perform badly. So I had a patient with a CTO, he's 60 years old, he has diabetes, and his CTO is in the circumflex. As you can see here, the circumflex is not a big vessel, it's lung disease, and you could, of course, put two long stents here and cover it up nicely. But you know, this is a diabetic patient, and he will probably have some most likely some problems in the future, because his diabetes will also not become better than rather worse. So in this case, again, I use a Wolverine balloon, I use an OPNNC balloon, and then I use 2 SELUTION balloons, 40 and 30 millimeters long. And this is what it looked like initially. Now, you would say, well, you know, initially it looks good, but in 6 months you will have late lumen loss like crazy, and you would have been much better with a stent. Well, I brought the patient back after 6 months just to also have some feedback for myself and for the patient. I told him that I'm using this now in a rather experimental way. We don't have big data to support this, but I said to him that I have a lot of data to support that long stents in diabetic patients do not perform well. And look what it looks like. After 6 months, it looks very nice. The patient hasn't had any restenosis, actually, and he's doing very well. And in fact, if you compare the angios immediately post-procedure and after 6 months, in fact, it actually looks better. The circumflex is wider. It has had this positive remodeling. So this encouragement, this case is now maybe about 7-8 months old since the re-angio, and this encouraged me to even push this further. Now, we did a lot of patients. This is just the summary of the abstract we just submitted for TCT. 61 patients, vast majority. We usually do intimal recanalization in Lucerne. We don't like the subintimal tracking. If possible, we try to avoid that. Most are treated with DS and DEB, but 20 patients of these are actually only with DEB, like in this patient. And we have only a 208 days mean follow-up. But target lesion failure rate is extremely good, 5.2% for this population. So I hope this abstract gets accepted TCT so we can share also there our experience. And the mechanism was actually in one patient, a non-healed dissection. This is a patient I treated. I had a dissection which I left behind, and then the patient came back after 4 months and had to put a stent because the dissection did not heal. And in only 2 patients we had true ISR, or let's say not instant because it was in a native vessel, but just in segments stenosis, if we go. Now, let's speak about some bifurcations. And bifurcations in the meaning that not treating the side branch, but bifurcation really considering the main branch, or trying to avoid stents in bifurcating lesions. This is a patient who has had a lot with his heart. This is a pericardial. He had constrictive pericarditis, and you can see this very nasty bifurcation lesion in the circumflex with the intermediate branch. I think if you try to stent here, you will end up putting 3 stents, and it will be difficult to solve this. You might get it to look good angiographically in the beginning, but this is bad because the patient has severe coronary disease because of he had radiation when he was a very young man with Hodgkin lymphoma. And he has had pericardectomy, and he has had TAVI, and he has a pacemaker. So really, this is not the patient you want to play with 3 different stents. So I actually treated him just with balloons, with 2 solution balloons, and this is the result after 4 months. And I think it looks good. It doesn't look perfect. But the patient doesn't have any symptoms. He will receive soon a CRT system because he's being continuously paced, but actually his angina disappeared completely. And I think cases like this somehow encouraged me to go further. Now, another difficult bifurcation. This is a case I treated last week. You have, and this was a patient who was a very close relative of a co-worker of ours. So it was really, I wanted to do my best for this patient. But I have this really bad lesion there, calcified. I have OCT, which I'm not showing here. And these 2 big side branches. Now, how can I do this case just to put a stent in and then save also these side branches? It's not that easy. You will surely appreciate that. Now, I pre-dilated again, cutting balloon. Then I used a 3.0 open balloon, and look what happened. The side branch is shut down. Now I gained the side branches. I put a small balloon in both side branches quickly because I had wired them, luckily, and I could establish flow in the side branches. But I said, I'm not putting a stent here. This is going to cause me a lot of trouble. So I said, why not use just a drug-eluting balloon? And I used a drug-eluting balloon, and this is what it looked at the end. I have nice luminal gain. The side branches are flowing. The patient's doing well. And I think in such a case, it's perfectly legitimate to try to do this. But unfortunately, we don't have data to support this. So I'm taking the risk, let's say, on my shoulders. But I had to do it because this was the best I could offer to the patient. Now, the last case I would like to share with you is a hybrid case. So this is a 69-year-old lady. She has severe LAD disease and severe RCA disease, as you will see. She did not want open CABG, so we offered her minimal invasive CABG, Lima to LAD, and said, let's do the RCA. Now, I would like to share with the RCA. The LAD was grafted 6 weeks ago. Now, here you can see there is a tight lesion in the mid part, but also very bad diffuse disease in the top part. And when you put a 6-French guide catheter there, it damped. So I had to treat that part as well. So what did we do here? Well, again, cutting it with a 2.5 balloon, then OPM balloon in the top part 3.0, in the distal part 2.5. And then I said, for the proximal part, the lesion was collapsing over and over. So I said, there in the ostial part, I will put a stent. because I want to have the ostium open because it was collapsing, the catheter was damping continuously. And for the distal part, I'll just use a balloon. And this is what I did. So the proximal part has a drug-eluting stent, and the distal part, I used the 40 millimeters 2.5 solution balloon. The important thing, maybe the last thing I would like to mention is, what I have learned so far with these cases I have done is to rather undersize. So if I use a 3.0 balloon OPN for luminal gain, I use a 2.5 solution balloon at very low pressure to just deliver the drug and try to seal those minor dissection you might have, because I think if you use, it's a semi-compliant balloon. If you use a too big balloon, then you dissect and then you will need a stent because you dissected with the balloon. So this is maybe a tip I can give to the people who are listening. Now, my take-home message is, I think, with careful lesion preparation, and we are not there at the end. I think this is the key before we establish these balloons even more in our clinical practice. We have to learn how to prepare lesions well. I think we are not there yet. My way, cut and crack, could be the way to go, but I don't have data for this, but it looks promising so far. And I think what I can say is SELUTION performs extremely well because the patients who come back, they have good results, and this gives me the hope that we can continue this way and we don't get any disappointment, hopefully, in the future. Thanks very much for listening. I hope I kept the time, and let's have a nice discussion to keep me awake.
Thank you so much, Flory. This was outstanding and real-world cases, right? We're not talking about focal in-stent restenosis or only side branches. I think this was very educative for all of us. So Flory, Ron, and Ricardo, we do have 3 excellent panelists that is gonna, I'm sure, bombard you with some questions. Aníbal Damonte, I already introduced. We have Marco Weinstein and Gaston Dussailan. So 3 very experienced interventionalists. I will give the word to them for questions, but just a quick question to Flory. This is already a good registry, right? With 400 patients, almost 500 balloons. How much late follow-up consistently do you have? And if you have some angiographic follow-up, this is already a paper because this is new technology.
Yeah, well, we also submitted the second abstract where we have a follow-up of about 9 months for the all-comers, and about 1/3 of our patients have had angiographic follow-up. And we are now summarizing all this data, and well, I will gladly suggest you as a reviewer if you don't mind. But I think we should really get out this data because it's not only me in the lab, but I have 2 outstanding interventional cardiologists working under me who just believe in this. And the only problem we have is that financially, actually, if the patient are staying overnight, then we are doing backwards. So in Switzerland, you are getting punished because you use drugs and balloons. So I have to withhold them and say, well, don't overdo it before the hospital administration notices this. But otherwise, it's patients are coming back, they're doing well, and this encourages you to use it more and more. But we will put this data out very quickly.
Wonderful. This reminds me, Ron, my fellowship with Gus Pichar that always said that better than any stent is no stent. So that will be interesting to see this new technology now with a different concept with lesion preparation that we didn't have so much before. So Aníbal, do you want to start with the first question?
Yes, for sure. Thanks, Alex. I just want to congratulate all the 3 speakers and Med-Alliance for organizing this symposium, because actually I think that there is with the available technology, there is an underuse of drug-coated balloon in regular practice. And I think that we will all agree, whether in Europe, the United States, Latin America, and maybe Asia, there is an underuse because the available technology does not, I think, does not have the results. We have not shown the results that we would like to see in our daily practice. This is completely out of the box because Ron clearly showed us the innovative technology that is behind this slow-release Cyrolimus And then Ricardo clearly showed us this amazing late loss, very low late loss for me in this first in man, and the cases that were presented by Dr. Kukuli were real world practice and very complex cases, most of them. So I would like to hear from you, how do you foresee the future of drug-coating balloons with this technology, with this new technology?
Ron?
Maybe I will start, and I will tell you that it's a question of believing and starting to do it, because the stent is very gratifying. You get a very good result. Some people even don't do vessel preparation, but we do pay a tax for that. I think that we have to start where the stents are not doing that good. So when you go to the small vessel, to the distal vessel, there is not even a question. I mean, we don't want to put stents in those areas. Then you have a side branch of bifurcation. So I think you don't start from the proximal LAD, the 3-5 vessel, although I think that fluorine does all of them. And I think you're going to get there if you're going to get after 400 cases. But how do you start? You start with those areas that you know that stents are not doing well. And I think we do have data today on small vessel, and we have, it's very intuitive that you can sometimes massage the distal vessel with balloon, but you want a drug-coated balloon. And the same for the bifurcation. So these areas, I think there is no question. You can go to other areas like diabetes. If you ask me, the future, in my perspective, is to go after the non-culprit lesion. And I will tell you one statement that I've been getting more and more fun of after the LRP study and the Prospect 2 study. Patients don't die from ischemia, but they die from rupture plaque. And those lesions that we would identify them, and now we have tools to identify as a vulnerable plaque, they're not obstructive lesions. So you don't really want to treat them with a stent, but if you want to passivate it, drug-eluting balloon could be a great application. And this is a whole new era, and you ask me about future. So my vision is that if we'll ever treat a vulnerable plaque, that would be the way to treat that, not necessarily a metallic stent. So it is a question of choice. If you want to get a very good result, no dissections, immediately larger acute gain, yeah, you can put the stent there, but there is a tax to pay. And I see those taxes because they still send me patients for brachytherapy, and those patients with 3, 4, 5 layers of stent. I see patients that they started with the proximal LAD, but they went all the way to the distal, almost to the apex. I mean, why do you do it? So in the US, we don't have drug-coated balloon for the coronary. And that's probably one of the reasons. But if you do have it, then you don't go beyond the mid vessel with a stent. You go with a drug-coated balloon. And my final comment, if you really want to see what happened in the periphery world, we completely moved from stents or POBA to drug-coated balloon. They are dominating right now the SFA. They are going to dominate also below the knee, which are small vessel. So there is not much competition there, I understand, but still they are better than drug-eluting stent. They are better than bare metal stent, and they're better than POBA. So the same analogy you can apply to coronary, again, maybe not 100%, but you have to start from somewhere. And I think one of the issues that bothered me all the years with drug-coated balloon was the fact that I didn't want to put paclitaxel in patients' coronary arteries. And no matter what you're going to tell me, it's a good and safe and it's effective, I didn't feel comfortable with paclitaxel in the coronary arteries because I've seen all the results with taxus and the aneurysm and the other toxicity. I think when you have a drug like Cyrolimus, it's a game changer. And if you can apply it and you get a good result, then why not? So I would start with a drug DEB, like the SELUTION SLR. If I have dissection, I will probably have to tackle that with a stent. My question to Florin, and I talked too much already, so I'm going to pass the question. What is your experience with those that you did the SELUTION and then you still were not satisfied with the result, whether it's a recoil or dissection, and you added a stent? Because then you have like 2 drug combination devices, drug-eluting balloon and drug-eluting stent?
Well, maybe before I answer your question, Ron, first my comment how I personally see the future. I'm of course younger than you and less experienced. In a way, I believe in a complete metal-free future. And I think maybe in about 15 years, we will be using proximally a scaffold and distally a balloon, a drug-eluting balloon. I think this should be our end vision for treating coronaries. I think putting metal in coronaries, I think this is not good for the future because, however, I mean, I pay a lot of attention to lesion preparation, to luminal gain. I use, in almost all my cases, intravascular imaging. I really do it with love. And sometimes when you see the patient coming back to 3, 4, 5 years, you still see a lot of luminal loss because this never stops. And that's why I think we should go away from that. Now, regarding the technical bit, how I solve that issue, it's always difficult. That's why I try to really decide if I should ever start with a drug-eluting balloon or just say, okay, well, this needs a stent. And for this, I think lesion preparation is the key, and this is really the focus for me for the next maybe 12 months to learn how to prepare a lesion. And maybe the trial which is planned by Med-Alliance, maybe it's a bit too early for that trial, because if the trial fails against stents, then it might fail because we have not understood how to prepare the lesion well, not because the balloon is not good. At the end, it will be a solution did not perform well. If it didn't, I hope it will. We will participate. But I think understanding how to prepare the lesion well. Now, there are cases where I've used, if I dissect with a drug-eluting balloon, that I use then another balloon for prolonged inflation, and then I put a second drug-eluting balloon on it. So I try to avoid this combination of one drug from the balloon with another drug from a Zion stent. But sometimes it's just not possible, and then I have to put another stent over it. It's not ideal, but I think we have to speak much more about how to prepare these lesions well for drug-eluting balloons, and when is enough luminal gain achieved, because very often that's the problem, that you try. If you want to achieve stent-like results, then you will dissect a lot. So you should be able to accept a little bit of less luminal gain acutely, but of course enough luminal gain for the patient to have no more angina and to be happy. And this is a difficult compromise, and I haven't understood it yet. I mean, I've used it hundreds of times, but I'm still working on myself.
Thank you, Flori. So, Gaston, do you want to make some comments and questions, please? I think we only have like 5 minutes to finish, so you and...
Yes, I have a comment. Well, thank you very much for inviting me in this exciting topic. I think one of the problems that we will have is that people are, especially young interventionalists, are not doing any balloon angioplasty. They think that balloons are just for preparing the way for the stent. So we have to teach them how to do balloon preparation, lesion preparation, dilatation, in order to have a good result and learn a lot about what is a good result with balloons, and then to apply the technology. That is really, really exciting. I have a question, if there's enough time, about the mechanism that this will produce a better lumen loss, less lumen loss. Is this inhibiting just the proliferation, or is also affecting the negative remodeling that is the main cause of restenosis after balloon dilatation? Is there any studies on that?
Yeah. So Ricardo, do you want to take this question and respond quickly, please?
Sure. I think both mechanisms are involved, and it's interesting to see in this first-in-man study there are 56 lesions, and only 12 were ISR. There are 44 lesions de novo. And the best results, angiographic results, were seen in the de novo, meaning that you can achieve a higher acute gain within the stent, but you're also going to have a higher loss. So the late run loss was higher there. With balloon angioplasty, as we know, we might have less acute gain, but less loss. So the percentage stenosis, the delta percentage stenosis was actually smaller in the de novo group. And just to make a comment about the discussion, I think my generation has been born, with stents. And I have to say that I'm becoming a believer as I'm seeing these results, and we start to do more and more cases with DCB. And I think it's really something that fellows from now on, physicians need to learn, like they learn how to do stenting for diffuse, distal, small vessels, and side branches.
Yes, it was a privilege to be here. Sorry that I was a little late. I was in another session and also stuck in the cath lab. So, Mike, actually, I think we have like 2 minutes. So I have 2 or 3 comments, Ronald. First, and I'll try to be very straight. First, we've been using drug-eluting balloons for a long time. We've been involved with some studies, and particularly every year involved in a session, but with paclitaxel-eluting balloons with Paratronic, as you know, one, because you're also involved with Paratronic as well. With their Magmaris, the Dreams, and all the sort of the bioabsorbable products. So we've been working together in this. I really believe in drug-eluting balloons, regardless of paclitaxel, and I also believe that Cyrolimus is a much more appealing drug, despite the fact that we don't know exactly the kinetics of Cyrolimus for DCB. I think SELUTION has a very particular one, and 90 days kinetic, very promising in lab, but we have to see in patients how it does. But my other comment is regarding lesion preparation. It has been stressed here, and we have a study. Robert Byrne and others have demonstrated in the desire that lesion preparation with cutting balloon is very, very important for using, especially for in-stent restenosis. We've been using, and I use it today in a case that the patient arrived in a non-STEMI today at the lab, and he had a tight lesion proximal LAD and a diffuse lesion distal LAD, and at the end, at the iris, it was dissection. It was what he had. So it's spontaneous dissection scat. So we were able to use a drug coating balloon. After we used the cutting balloon for the scat, we used drug coating balloon for the whole distal LAD today. This was today. Unfortunately, I cannot show you the images. And then the patient came in for acute MI after. We opened the lesion, and then put the stent as usually, and after post-dilated the stent, no flow. So this is another indication for drug-coating balloons. Acute MI. Bokal in South Korea has a very, very large experience and a registry with acute MI. If we use drug-coating balloons for acute MI standing, I know it seems very, very tricky, we can prevent no reflow. So this is another indication besides its small vessel, diffuse lesions, in-stent restenosis. Bifurcation we use a lot for when we have in-stent restenosis in bifurcation in order to prevent using a third or second layer of stent. This is a very, very solid indication for DCB. I think there's a lot of indications for DCB, and hopefully the Cyrolimus platform with SELUTION will be even better. Thanks a lot.
Thank you, Marco. The final comments is from your side, Ron, please.
Well, this has been an exciting symposium, and we still learn. There is so much to learn on drug-coated balloon, and especially on Cyrolimus. But I'm impressed by the data that Ricardo showed. I think this is not like anecdotal. We used to know that give me results of 20 cases, and I'll tell you how the 200 is going to look, right, Alex? So you gave us 45, 50. I do concur with you that the de novo is more important than the instant restenosis, because it will use less stents, we're going to have less instant restenosis. And also, instant restenosis of drug-eluting stent is not that trivial to treat. It's not that simple. The novo lesion is much more easier. And finally, I have to throw here imaging, because I think imaging is always going to help us to verify that we don't, that we prep the vessel properly. Just throwing a balloon and inflating 2 millimeter, 2.5 on a 4 vessel, that's not vessel preparation. We really have to demonstrate that the vessel preparation was done. So we are in an era of an exciting technology. We have to gradually move it on forward. One thing that I commend Med-Alliance is that they are not shy of clinical trials. That for a startup company to throw 3,300 patient study, 500 patients in instant restenosis, that's huge, huge undertaking. So I wish them that the study will be positive. I'm optimistic that they would, from everything that I saw so far, but all remain to be seen. So continue the research and the follow-up of those patients, and thanks for everyone.
Thank you so much. Thank you, SOLACI. Thank you, Med-Alliance. Thank you, the speakers and the panelists. Have a great night and a good morning for you, Flory. Bye-bye.
Thanks a lot.
Bye.
Thanks.
Bye. Thank you.
Bye.